People are still trickling in.
Hello, everybody. Welcome to the monthly webinar for the National Center for Genome Analysis Support. My name is Robert Ping, and if you have any issues, please reach out to me in the chat during the talk today. And I'm going to let Tom Doak take it away.
Cool. Hello, everyone. This is the monthly NT-GAS webinar. It's hosted through the Supercomputer for Everyone series at Indiana University. Just a second. There we go. A few things I want to remind you of, a handful of things. How to get hold of us, how to interact with us. So along the bottom here, we have ntgas.org. That's our website. Has a lot of good information. We have a pretty active blog series having to do with bioinformatics and using HPC systems in general. We have a discourse help desk. Right. There we go. We have a Discord help desk. This is checked every day. But in particular, we have live office hours the second week after this. So it's the fourth Tuesday at 2 o'clock. And several of us, the NT-GAS team, will be there and happy to answer questions. YouTube channel, IU_PTI, has a lot of videos of ours. Always, you can write the help desk at help@ntgas.org. And we're really pretty active on Twitter as well. So feel free to follow us. Okay. Next month, before I introduce Jean-Francois, we will have Lydia Bright from SUNY New Paltz, uncovering molecular determinants of holospor infection in paramecium cells. So paramecium are a single-celled eukaryote. They suffer infections by bacteria just the way everyone else does. And she's interested in what those responses are. Jean-Francois will give, assistant professor at Mississippi State University, will talk about a genome-wide view of transcription fidelity. He did his undergraduate work in computer science at Paris 5, then moved on to a master's in bioinformatics at Paris 7. He was PhD in the University of Lyon, working on the genomics of Paramecium whole genome duplications. He was a postdoc with Michael Lynch when Mike was here at IU, and Jean-Francois moved to ASU when Mike moved. So he's been with Mike in both places. Since 2018, he's been an assistant professor at Mississippi State University, Department of Biological Sciences. Just a few credits here. NT-GAS is affiliated with Indiana University, Pervasive Technology Institute. We are a center within the Pervasive Technology Institute. Within IU, we're a management unit of Research Technologies, Division of University Information Technology Services. And the NT-GAS grant, NSF funding grant, is a collaboration between us, IU Pervasive Technologies Institute, and the Pittsburgh Supercomputing Center at Carnegie Mellon University. And with that, I'm going to stop sharing. And Jeff, go ahead and share.
Okay, thank you very much, Tom, for the introduction. Can you hear me fine?
I can.
Yeah, okay, thank you. So let me just go in full screen. So just a quick heads up. If anyone has a question during the talk, if there's something that's really not clear, feel free to unmute yourself and ask the question. But otherwise, if it's more like a general question, just keep it for the end. But if there's something that really is not clear, please don't hesitate to interrupt me. And I will try to be as clear as I can, but I know I'm aware I do have a little bit of an accent, so sometimes it can be difficult to understand what I say, especially over Zoom. And I might be a little bit cranky today because I was trying to solve actually some trouble on our own little supercomputer this morning, and it's still not solved. So I'm gonna try to stay positive during this whole talk. And as Tom mentioned, I was a postdoc at IU in the Lynch lab. So a lot of this work was actually started when I was a postdoc. It's work that I continue doing, and it's work for which I've used the computational resources at IU a lot. So the talk is a genome-wide view of transcription fidelity. And just to remind you the basics of biology that's behind this, we've all learned pretty early on, probably in high school, that there's something called base pairing, where you have guanine that pairs with cytosines and adenine that pairs with thymine. So we've all memorized that probably at some point in high school. And this base pairing is at the base of the most fundamental processes in the cell. So replication works using this base pairing. Transcription also uses it. Translation uses base pairing to pair the tRNAs with the messenger RNA. And all these processes are incredibly accurate. You know, the replication, for example, makes a mistake in base pairing at a rate of about 2 × 10⁻⁻10 per cell division per nucleotide. So it's extremely accurate, but still there are errors that happen sometimes when the base pairing is not done correctly. And what happens when these errors occur? Well, these errors are very important, and that's why I'm super interested in looking at what's happening when these errors happen, because these errors are what actually make mutations. And without mutations, there would be no evolution. We wouldn't be here on Zoom today trying to understand cellular processes and using supercomputers. So that's one of the reasons I'm interested into this. There's been a lot of research on, first of all, the rate at which mutations happen in the DNA. Also, a lot of research on what are the consequences of these mutations. But until recently, there was very, very little research on the rate of transcription errors, of errors that happen when the RNA polymerase make RNA molecules. So just to make sure we are on the same page, what do I mean by transcription fidelity and what do I mean by a transcription or a transcript error? So that would be simply a difference between the RNA molecule and the DNA molecule from which the RNA was transcribed. And these errors can come from 2 possible sources. One is simply a misincorporation by the RNA polymerase. So during transcription, if the RNA polymerase is reading this blue strand here as the template, and it's positioned in front of a G, it's supposed to incorporate a C in the new RNA molecule. But sometimes it's going to make a mistake. And in this case, for example, it incorporates an A. So that would be what I would call a C to A transcript error. It's also possible sometimes that the RNA polymerase does a perfectly good job at transcribing, but then after transcription, there can be some damage happening to the RNA molecule, which will replace one nucleotide for another. So why should we care about transcription errors? Well, we have reasons to think that whenever transcripts that contain errors are produced in the cell, they can be translated which can lead to the production of non-functional and possibly misfolded proteins. And these misfolded proteins, they can aggregate in the cell. They can cause a lot of trouble for the cellular health, and that could be the cause of some diseases. So we're interested, among other things, to investigate how transcription fidelity could be linked to some human health disorders, and especially to some aging disorders. And there's been some very recent work that we're now starting to be involved with that linked some human disorders, some really like terrible diseases with mutations in the RNA polymerase, which would now shown are error prone. So, okay, if we're interested in looking at the fidelity of transcription, the first thing we need is a method to detect transcription errors. So how are we going to do that? Well, the first idea, which I will call the naive approach, will be to simply use regular RNA-seq data. And I assume everyone here is somewhat familiar with RNA-seq. It's just a method in which you extract RNA from the cell, you reverse transcribe the RNA into cDNAs, make a library, sequence it. It's typically used to get information about expression level of genes, to also annotate genomes. And here you will think that, well, if there were to be a transcript error, it will show up as a mismatch between your RNA-seq read and the reference genome when you align the RNA-seq read to the reference genome. So that's what it will look like. You will build your RNA-seq library, get the sequencing reads, align the reads to the genome, and here this little red bar. I don't know if you can see my my mouse. I hope you can see. Maybe I can use another pointer, but hopefully you can see my mouse where I'm pointing at. So there's this little red bar here that is supposed to represent the position of one transcription error. And that will show up as a mismatch between your RNA-seq read and the reference genome. So maybe at this position, you're expecting to see a G and one of your reads as an A. That will be a transcription error. Now, as I said, that's a naive approach, because the problem with that is most of the mismatches that you're going to find between the RNA-seq reads and the genome will come from sequencing errors. So the sequencing technology that you're going to use to sequence your RNA-seq library is also going to produce errors. Whether you use Illumina or PacBio or BGI or any sequencing technology that you like, none of them is 100% accurate. And even though the error rate is pretty low, typically for Illumina, if you filter your data for only high quality base calls, you're still going to have about 1 in 1,000 to maybe 1 in 10,000 if you're really strict base calls that are erroneous. And that's going to be 10 to 100 times more than what we suspect the actual transcription error rate is. So something like 99% of the mismatch that you find between your RNA-seq read and your genome are actually sequencing errors and not transcription errors. So if you use this approach, it's like you're trying to find a needle in a haystack, your pile of hay being all the sequencing errors and the needle being the one rare transcription error that you're trying to find. Now that wouldn't be too much of a problem if this analogy was correct, because if you use big supercomputers like the one that we have access to, you could just take a lot of time to sort out the hay and find the needle. Or if you're smart, you just burn down the haystack and only the middle needle will remain in there. The real problem here is that our needle looks exactly like a piece of hay. Like, how do we tell apart the mismatch that comes from a transcript error from the mismatch that comes from a sequencing error. So that's what I was working on when I was a postdoc in the Lynch lab. And we had a lot of really useful discussion, including with Tom and Matt Ackerman and a few others. We came up with a method to do that. And then some other people came up with a much better, much smarter method. So I'm going to present the much smarter method, which I did not invent. I just contributed to adapting this method for this specific project. So the solution is we're basically going to try to sequence the same RNA molecules multiple times. And we're going to use the redundancy to sort out sequencing errors from transcription errors. And we do that with a method that's called rolling circle, which is a method that we actually borrowed from the field of virology. So the way it works basically is you want to make turn your RNA molecules into circles and then use the reverse transcriptase to reverse transcribe your RNA molecule. And because it's a circle, the RT is going to go around the circle multiple times and create these long cDNAs, which are made of tandem copies of your initial RNA fragment. So I'm going to walk you a little bit more slowly through the whole process. So the way the method works, you will start by extracting the RNA from whichever organism that you are studying. And these little things here are supposed to represent RNA molecules. You can see that my artistic capabilities are not that great. And this little red star here represents the position of an actual transcription error. So the first step then will be to, the next step will be to fragment the RNA molecules. We want to make fragments that are about a third the size of a typical sequencing read so that we can get 3 repeats of each fragment in our sequencing. Then the fragments are circularized. So you use a ligase to ligate the 2 ends of your RNA fragment. And then that's when you do the reverse transcription. So your reverse transcriptase is going to go around the circle again and again and again and make these long cDNAs. The beauty of this approach is that if you had a transcription error somewhere in your initial RNA molecule, it's going to be present in all the copies of your initial fragment, while the sequencing errors are going to be randomly distributed alongside the sequence. So when we sequence these long cDNAs, then we do a bit of bioinformatics magic. We find the repeats and we can stack the sequence, the repeats like that. If we find a position where all the repeats here support a certain mismatch to the genome, we're very confident that this was something that was present in the RNA molecule to start with. It would be very unlikely to have the same sequencing error happening 3 times at the same location. So that's the basic idea of how we sort out sequencing errors from actual transcription errors. Now, normally when I present this, I skip this part, but for today, I think you might want to know a little bit more about exactly how this analysis pipeline works. And in just a little bit more details, the way it works is I will start by doing a very classic step of every RNA-seq analysis, just trimming the sequencing reads. So to remove the ends that are low quality, and also to remove the primer sequences that can be present in some of them. And then I will identify the repeats inside each read. So the reads are supposed to be made of multiple tandem repeats of a given sequence. And for that, I wrote my own C++ programs, which I'm currently in the process of updating so that I can use OpenMP and parallelization to make things go a bit faster. So this here will show you the sequencing read that has been organized so that you can see the repeats. And you have the first half of it, for which we have 3 repeats, and the second half that is covered by only 2 repeats. So here at this position, for example, you can see that you have 2 repeats that tell you there's an A, and the third repeat that says it's a G. Well, the most likely explanation for that is that this G here was a sequencing error, or possibly a mistake made by the reverse transcription. So what I do with this sequence is that I first compute a consensus sequence, which is basically finding at every position which nucleotide is the most likely one to represent the nucleotide that was in the RNA molecule. So when you have the same base call at all the positions, it's very easy. Here you have all 3 base calls are for a T. So I say my consensus is a T. When you have discordance between the repeats, for example, here you have 2 A's and 1 G's. The way I do that is basically I will just do the sum of quality scores for the 2 A's and then compare it to the quality score of a G, and whichever one is the highest will become the consensus. Yeah. So now I have this consensus sequence, which hopefully represents the sequence of my RNA fragment, my RNA molecule. However, there's a small difficulty here, which is that when we do the reverse transcription on these circular RNAs, the reverse transcription can start anywhere on the circle. We use random primers to prime the reverse transcription, so it does not necessarily start at the position which was the end of the RNA fragment. So here, this C that's in blue here is representing the 5' end of the initial RNA fragment. And this other C here will be the 3' end, and that's where the 2 ends have been ligated. And in this example, the reverse transcription started here, roughly halfway through the molecule at this T. So when I going to identify my repeats and find my consensus sequence is going to start here at this position, which is going to cause some difficulties when I try to map this consensus sequence to the genome, because the first half here is going to map contiguously somewhere, and then the second half of the sequence is going to map upstream of it. And mapping programs don't really like dealing with that type of kind of like chimeric sequences, and they're not very good at mapping them. So I was trying to find a solution to how to solve this problem, and the solution was given to me by Laurent Duret, who was a PhD student in the lab at the time. And there's a little trick that we use, which is that we do a concatemer of our consensus sequence. So we use 2 repeats of it, so that somewhere in this concatemer, there will be the full uninterrupted sequence that has to match to the genome. And then we can use this with BLAST to find where this comes from on the genome. And again, I apologize for the quality of illustrations. I had a big plan to make something a lot better this morning, and had to deal with some other issues with the server. So once I figured out the correct order of the nucleotides in my consensus sequence, so I figured out which nucleotide is supposed to be the first one in the RNA fragment, I can align it to the genome and look for every mismatch that I find between the sequence and the genome. And because I keep track of the data in all the repeats, I can call transcription errors specifically at positions where I have very high confidence in my base call. So that will be positions that are covered by at least 3 repeats, and for which each of the repeat has a high base call quality score. So here, for example, I will have one such transcript error at this position where I have a T that is mismatched with a G on the genome, and that would be a G to U or a G to T, depending if you want to talk in RNA or DNA alphabet, transcription error at this location. So we've applied this technique first on yeast, and that was work that I did when I was still a postdoc in the Lynch lab, and I was working with a PhD student. I was also very heavily involved in developing the technique, Wei Li, and Mark Vermust, who is now at University of Southern California. So the 3 of us would work together at the bench to generate the data, and then I will do the analysis that I just presented. And with this method, we very quickly managed to build databases of hundreds of thousands of transcription errors. So the nice thing with this method is that you basically find transcription errors all alongside the genome everywhere. Like everywhere that you can get RNA from, you can find transcription errors. So we have a really nice coverage alongside the genome. That's a circ plot of the yeast. You have the yeast karyotype, and each dot here represents the position of one transcription error that we found with our pipeline. And you can see that we find some everywhere. You have the details alongside one chromosome. And then we can start looking at the actual error rate, see how frequently these transcription errors happen. And if you remember Bio 101, there are different RNA polymerases in eukaryotes. The one that we're going to start with is RNA polymerase 2, which is the one that makes the messenger RNAs. So they're the ones that transcribe protein coding genes. And in this data set in yeast, we found in excess of 15,000 transcription errors in messenger RNAs for an error rate of a little bit less than 4 × 10⁻⁶. So that would be the rate of base substitution per nucleotide transcribed. So if you want to see the glass half full, you can say, well, that's only one error for every 250,000 nucleotides transcribed. So that seems pretty accurate. It also translates in about one in every 150 transcript that contains at least one error. Now, if you want to see the glass half empty, you will compare it to the DNA mutation rate, which is 4 orders of magnitude lower. So this process of transcription is a lot more error prone than the process of replication of the DNA. Now, if we look at the RNA polymerase 1, which is the enzyme that specifically makes the ribosomal RNA molecules, it has a very similar error rate to that of RNA pol 2, again, on the order of 4 × 10⁻⁶. We also have an RNA polymerase 3, which is its job is to transcribe the tRNAs and also a certain number of non-coding RNAs. And this one has a much higher, more than twice as error-prone as the other one. And there's also an RNA polymerase that is specific to the mitochondria, and this one is also fairly error-prone, for we don't have a lot of coverage for these regions. Now, the main advantage of our method too is that not only we can get the rate, but we can see each 12 of the possible errors, how frequently they occur. And what we see is that for both RNA pol 1 and RNA pol 2, the most common type of error is a C to U, meaning that we observe uracil in the RNA at a location where we were expecting a cytosine. So we're still working on trying to understand why these ones are so frequent, and I'm happy to talk about that at the end. But that's the pattern that we see for both RNA pol 1 and RNA pol 2. Now, if we add RNA pol 3 to this graph, we see a very different pattern where G2A errors here are the most common type of errors made by RNA pol 3. So this one is less accurate, but it's not that it's making more error, a little bit more of all possible errors. It's mostly that it makes a lot more of one type of error, G2A. For some reason, it looks like RNA pol 3 is pretty bad at this specific type of errors. And then the mitochondrial RNA polymerase, its increased error rate is mostly driven by an increase in A to G errors. So the next thing we did was look at what's going on when we start to poke at the RNA polymerase. So the RNA polymerases are reasonably complex enzymes made of 12 different subunits. And there are some subunits that are known to be, you can knock out the subunit and you still have a viable individual, at least in yeast. And we wanted to look at what happens to the fidelity of transcription when we start messing around with the RNA polymerase. So we looked at a couple of different mutants. One, which is a full knockout of this little subunit in orange here, RPB9. There was already data indicating that it was probably involved in the fidelity of transcription. So we looked at what happens when we knock out this subunit in yeast. We looked at a point mutation in this subunit, RPB1. So this one, RPB1, is the largest subunit of RNA polymerase. And we can't do a full knockout that completely abolishes transcription, and that is not viable. So one amino acid substitution at this specific position in RPB1, and then also a knockout of a general transcription elongation factor called DST1 in yeast. And if you work on transcription in humans, you might have heard of it under the name TF2S. So all three mutants showed a very strong, very significant increase in transcription error rate compared to the wild type. And all three mutants are specific to the RNA polymerase 2. All these subunits are specific to RNA polymerase 2. So when we looked at the rate for RNA polymerase 1, we saw that the mutation didn't do anything for this, which was a nice control that told us we didn't just completely mess up the sequencing for the mutants or something like that. But we also looked at what happens when we knock out the homologous subunit in RNA pol 1. So we did that for this RPB9 subunit in pol 2. It has a homolog in pol 1, which is called A12.2. So we knocked out this subunit. And this time, that's the data that's in pink here, we see a very strong increase in error rate, specifically for the RNAs that are made by RNA pol 1. So what we did then is we looked at what type of errors are increased in frequency in these mutants. And the pattern that we saw that is extremely strong is that for all three mutants, most of this increase in error rate is driven almost exclusively by these G2A types of errors. It looks like for some reason, as soon as we disturb the RNA polymerase, it starts making a lot of G2A errors. So it's very bad at reading the cytosines and incorporating a G opposite the cytosine. For some reason, it tends to put an A there. And that's reminiscent to the pattern that we observed for RNA polymerase III. So right now I'm trying to work on trying to understand what is the cause of this pattern. So just to illustrate what a G to A error is when there's a C in the DNA template and the RNA polymerase is incorporating an A in the RNA instead of the G that it should do if it were to follow the normal base pairing rules. So one hypothesis is that cytosines, we know they are very prone to damage. So could it be that when there's a damage to the cytosine, the RNA polymerase normally has a way to deal with it, but that these mutant RNA polymerase just don't really know how to deal with the damaged cytosines and incorporate an A opposite of that. So
you want to pause for just 1 second? You have a question in the chat as well.
Oh, yes. Yes.
It says the C to U transition seen in RNA-PI and RNA-PI is also a classic signature of damage in ancient DNA due to deamination of cytosine to uracil. Is it possible that the cytoplasmic environment is more prone to this reaction than the nuclear environment? And I'm reading that out loud so that people who are watching the recording later know that the question is, what the question is that you're answering.
Okay, yeah, thank you. So it's very possible that the, I'm just gonna track back a little bit, that this pattern here, this high rate of C to U's that we observe for wild type RNA polymerases could be mostly cytosine deamination, so some damage that happens to the RNA molecules rather than misincorporation made by the RNA polymerase during transcription. And we're trying to figure out the best ways to test this hypothesis, which is not very easy. One way would be to try to capture specifically the nascent RNA molecules so that there will not have been a lot of time for deamination to happen. So if you could capture the nascent RNA in the nucleus, we might observe a much lower rate of C to U, and that would fit with the hypothesis that was presented in this question. So I hope that answers the question. And then here, what could happen is there could be a C to U deamination on the DNA template strand. And if that were to happen, then you would expect the RNA polymerase to start incorporating an A instead of a G at this position. And let me check in the chat if there's, okay, thank you. So one thing that we looked at is also what happens when we actually start increasing the amount of damage to the DNA. So a lot of, the FDA and EPA, they test products before they're approved, and there's a lot of research on whether certain chemicals are mutagens or not. And what these studies do typically is they look at the effect of a chemical on the mutation rate. But what if some chemicals did not really increase the mutation rate, but increase the transcription error rate? That could have some significant consequences for human health, and we would miss these problems with the method that we had up to date. So one such chemical that we looked at is methyl nitronitrosoguanidine, which I would just refer to as MNNG, which is a very well-known mutagen. It increases in yeast in the concentration that we used it. It increased DNA mutation rate about 3-fold, but it increased transcription error rate more than 20-fold. So what we found is that there has to be a lot of damage to the DNA, which is made by these chemicals, that will eventually be repaired before or during DNA replication. But before they're repaired, there's opportunity for this to be passed on into the transcripts, and you could end up with producing a ton of error-containing transcripts because of exposure to these chemicals. So that's trying to go a bit fast here to finish in time. So that's what we observed here when we exposed yeast to MNNG. We see the error rate of transcription that increases about more than 20-fold. And then if we let the yeast cells recover for a couple of hours, we see, at least for RNA pol2, the messenger RNAs are made new more frequently than ribosomal RNA. We see the error rate that goes down. So there's some recovery. So we think what happens is that some DNA repair is happening, and then it's preventing these errors from being made again. And then when we did the same experiment, but we knocked out the gene MGT1, which is one of the genes involved in DNA repair, we see that this recovery that is very strong here in the wild type is now much weaker. So just very briefly in the last couple of minutes, here everything I presented was looking specifically at base substitutions when the RNA polymerase incorporates the wrong nucleotide. But just like with mutations, we also have another type of errors, which are insertions and deletions, which are referred to as indels. And similar to what we see for mutations, the indel rate for RNA polymerases is about 10 times lower than the rate of base substitutions. And we also observed that all of our mutants that had elevated base substitution error rate, they also have an elevated indel error rate. We also observed that the probability of an indel increases with homopolymer runs. So if the RNA polymerase is transcribing a stretch of DNA which reads AAAAA, then the probability of making an error is much, much higher than if it's transcribing a region without such homopolymer runs. And we do have reasons. We have some understanding of why that is. Whenever it makes a mistake, the RNA polymerase has to backtrack and realign the nascent RNA with the genome sequence with the template strand. And during this realignment process, it can very easily shift one base upstream or downstream when it's on a homopolymer run like that. And then a very quick story about, maybe I'm going to skip that because it's already 1:45. So I'm going to just skip this little story about errors that produce a stop codon in the transcript. We can see that these things they happen and that they are removed by a cellular mechanism called NMD, which removes transcripts that contain premature termination codon. But if you're interested in that, I can give you more information on that during the questions. But I want to keep a little bit more time for questions, so I'm going to skip that mostly and just tell you that we started this working on yeast because it's very convenient. It's well-behaved model organism, easy to grow in the lab, small genome, so not putting too much stress on the supercomputers when we do the analysis. But now we have data for also nematodes, Drosophila, mice, and humans. I don't have humans on this slide, but we do have transcription error data for a large number of organisms now. So just conclusions. If you've never heard of it, this method, rolling circle, it's still fairly new. It's a very efficient method for to get a genome-wide view of transcription errors. Let me see, there's something in the chat. Did you consider other methods besides circle sequencing for getting multiple reads from the same transcript? for example, DAPSIC, and are there reasons that circle sequencing is preferable? Yeah, another great question. So we did, first of all, we tried another method that is pretty much the same as something that people use when they do single cell transcriptomics, where you use a unique molecular tag. You tag your RNA molecules with this tag, and you use that. The main problem of all these other methods is that the way they generate for redundancy, your different sequencing reads that come from the same RNA molecule, they are not physically linked. And you end up having to sequence extremely, extremely deep before you finally get multiple reads that come from the same RNA molecule. Really, the big advantage of the rolling circle is that the repeats from your initial RNA molecule, they are physically linked. So pretty much every single sequencing read that you generate is going to be informative. So it's for this approach, we find the rolling circle to be a lot more efficient than any of the other methods that is available. Okay, so back to the conclusions. We have a pretty good view of transcription fidelity for a couple of mutants of RNA pol 1, 2, and we're now working on mutants of RNA pol 3. So trying to figure out what are the molecular mechanisms that are responsible for the fidelity of transcription. We see a generic trend that all these mutants have an increased G2A error rate. To be honest, it's a pattern I noticed. Like Tom probably saw me present this when I was still a postdoc in the Lynch lab, and we're still working on figuring out what is the real molecular mechanism that is responsible for this pattern. And I hope to have some breakthrough this summer. And one area of research that we're also investigating is how does DNA damage will impact the fidelity of transcription. And I'm just gonna say a few words about a really cool application of this method, which I didn't envision early on. As I said, the rolling circle was initially developed by people who were interested in looking at mutation rates in viruses. So this group, Ashley Acevedo and Raul Andino, they're the ones who should be credited for developing the rolling circle method. And they were interested in getting very high accurate sequencing reads for viruses. And then one day, Chi-Chu Chen was a PhD student in the lab, saw this paper and brought this paper to my attention. And that's where we're like, well, we should use this for our transcription errors. So we made some modifications to this method, but that's where the idea of a rolling circle came from. And last year, I started a collaboration to bring back rolling circle to its initial role. We are now using it to try to measure the mutation rate of the SARS-CoV-2 virus. You might have heard of it. There's something called coronavirus that's been causing some issues in the past year or so. And there's a lot of research ongoing on that. And we now have a big collaboration with people in the Netherlands that have a BSL-3 lab. They are in charge of growing the virus. And then we are doing rolling circle on the viral RNA genome, because this virus, SARS-CoV-2, is using an RNA genome. So we can directly use rolling circle to get 100% accurate sequencing of different, of all the population of viruses, and deduce the mutation rate of the virus. We can also use this to estimate the fitness consequences of different mutations. So that's an ongoing project. I wish I could share some of the early data with you today, but we're still a little bit too early in the process. It took a very long time to get the project running, and I don't really have a lot of data to share with you now, but hopefully there will be some exciting SARS-CoV-2 research coming out of that. And with this, I'd like to thank all the people who were involved in that. So there are lots of people in the Lynch lab who were with whom I had lots of very useful discussion, and that includes Tom, Matt, and a few others. But Wei Li is the PhD student who was really heavily involved in that, who did an amazing work to get the method to work at the bench. Mark Vermulst, who also had some really good ideas on how to improve the method. This work that I presented is funded by NIA and from NIH. So we're interested in trying to figure out how transcription errors can play a role in aging. So we do have a grant on that. Right now, I'm supported by Mississippi State University. And I don't have, I guess I should have added NCGAS on that. A lot of the early computations and development for the method was done on the servers at IU. And with that, thank you for your attention, and I'd be happy to answer any question you may have.
Hey, it looks like you do have another question in the chat there.
Yeah.
So before we start on that, let me just remind everyone that this was recorded. And after a bit of processing, we will get it up on YouTube, and we'll send out an announcement when that happens. Okay, back to questions.
The question on the chat is, if you are using poly-A selection to capture mature mRNA, not sure if you are or not, could you use this selection to instead get rid of mature mRNA? Essentially, leaving behind mRNA that hasn't been capped / tailed yet, and so will still have been present in the nucleus. Just thinking again about possible environmental differences and resulting RNA-P mutation rates inside versus outside the nucleus. So we do use a poly-A capture to get mostly messenger RNAs. These are never 100% efficient. We always have some ribosomal RNA and tRNAs that that remain in the mix. Now, I guess what you're getting at is what would be the best method to capture the very young RNA molecules when they're still in the nucleus. And I know there are a couple of methods. There are methods to capture the nascent RNA molecules, but I've never actively used them in the lab, so I'm not very familiar with them. And if you do have experience with that, Matthew, I'd be happy to keep chatting with you if you have some. I'm always happy to take some good advice on how to do that. If we were to simply get rid of the poly-A selection, we would just get a ton of ribosomal RNA. So we need to do something a little bit fancier. So I will need to look into more details about how the methods that capture nascent RNA molecules, how they work. I forgot how they work exactly. And
any other questions?
Feel free to unmute yourself if you want to ask questions. I mean, I'm happy to read the questions in the chat, but you can also just unmute yourself and ask questions directly.
Yeah, I have a question or a couple. They might be a little naive because this is a little outside of, but so I was thinking about like the specific kinds of errors, like G to A and C to U and things like that. And I'm curious if I guess how often the same transcripts like produce errors? Like, are there like sequence motifs or secondary structure kind of things that kind of predispose these kinds of mutations too?
Yeah, no, that's another great question. So we looked at a couple of things for that. The first thing the first information I need to give you there is whenever there is a position in a transcript where we find errors again and again and again, we typically throw this out because this could be caused by mutation in the DNA, especially when you look at a deep read organism. If your organism is heterozygote at a given locus, you might have half of your reads that will have something that looks like an error, but it's just that the DNA was different from that of the reference genome that we use for the analysis. So whenever we have a really high error rate at a certain position, we exclude this. Now, we did notice that the nucleotide context influences the error rate. So some errors, I forgot which ones, but I think probably the C2Us and the G2A, they are the rate at which they happen is going to change depending on the upstream and the downstream nucleotide. So I unfortunately don't have this slide here, and I don't remember on top of my head. But for example, if you have like GCA, the error rate of the C is going to be different as if the context were AAC, for example, something like that. So we do see that the local nucleotide context influences the error rate. As far as the structure, we are only starting to look into that now in specifically in the ribosomal RNA, because there's a lot of information about the structure of a ribosomal RNA. And one thing that's possible is that some secondary structures might be more prone to recruit some of the RNA editing enzymes, and that there could be some post-transcriptional modification that can happen at very low frequency, which we would see as transcription errors. So that's something, that's an area of research that we are actively looking into.
So following up on that, do you think that, if you looked across a gene family where you have like, newly diverged things, for instance, like you find anything interesting with like error rates there versus like very divergent sets? And this is kind of a separate question, but you mentioned heterozygosity complicates things, but one question I had there is if you think that highly heterozygous things generally tolerate a lot more of these kinds of errors.
Okay, so for the first question, I guess the answer is that we didn't see a lot of changes in error rate on a gene-by-gene basis. But we don't really have a whole lot of statistical power to detect this. So it's not very easy to answer that question now. Now, as far as the heterozygotes, for us, we see that mostly as a technical complication. We want to make sure that when we find like the rolling circle, basically you can see that as a way to get the sequence of the RNA with 100% accuracy. You get rid of all sequencing errors and all mistakes by the reverse transcriptase. But then we need to make sure that what the sequence of the DNA was at the locus that was being transcribed. And that's where the heterozygote things become a problem, because some of the mismatches that we observe might not be transcription errors at all. They might be just because the organism is a heterozygote, or it could even be a somatic mutation. When you do that on organisms, like if you do that on nematodes, maybe you have a mutation in one cell out of the 1,000 cells that this nematode is made of, And whenever you get an RNA that comes from this cell, you're going to match it to the reference genome. You're going to see the mismatch. You're going to think you're looking at a transcription error, where in reality, you're looking at a somatic mutation. So for me, I see these as more like technical difficulties to solve for the method. And I didn't really think whether organisms that tend to tolerate higher level of heterozygosity, if they also tolerate higher error rate of their RNA polymerases. That's definitely an interesting idea to look into. One thing I can tell you is that something that was surprising to us is that there's not a whole lot of variation in these transcription error rates between organisms. From yeast to humans, that's not a whole lot of variation. Nothing like the level of variation that we observe for DNA mutations. So I would be surprised if there was really a strong correlation between how tolerant some organisms are to a high level of heterozygosity and the tolerance level to transcription errors. But that's an interesting idea to look into.
Thanks.
Any more questions? Well, let's thank Jean-Francois one more time.
Well, thank you all for attending. Thank you for a great set of questions. And if anyone has questions or want to, has ideas we want to chat about, feel free to reach out by email. I'm happy to talk about this anytime. But thanks a lot.
Thank you.
All right. Bye, everyone.